have to explain to you how we actually get these hormone cycle data from the animals. In particular, what we're doing nowadays from mice, these older data, if I go back to 1 year, so here these are, of course, also cycled animals, but there we were doing that with rats. And in a rat, you may imagine it's a relatively big animal. There's no problem over a period of 3 hours or so to withdraw about a milliliter of blood every 20 minutes, spin down the hematocrit, resuspend it, and re-inject it where you keep the plasma for later analysis of hormone content. If you did that to a mouse, you'd kill the mouse. So how do we do it here? So we based ourselves there on a paper from the Chen lab in Brisbane, Australia, who developed a really highly sensitive ELISA method that allows to determine plasma hormone levels, protein hormone levels. And in our case, we're using it for growth hormone determination based on minute amounts of plasma. So what the whole method entails then is, if you want to do that, it involves handling of the animals as opposed to what we were able to do with rats. A mouse is also too small to give it a permanent access from which you can just withdraw. So when you want to withdraw blood, you have to handle the animals. So you have to for a really long time, habituate them to the handling, because any form of stress will result in the abolition of the rhythmicity of hormone secretion. So that is the step. And then in our case, day 21, when they are habituated, for every 20 minutes over a long period of time, you collect 4 microliters. You can go down to 2 microliters of blood every 20 minutes. And that is enough plasma then in that little drop of blood to determine our growth hormone level using the so-called sandwich ELISA, which is based on a capturing antibody that is very highly sensitive and selective for growth hormone, and then a signal amplification using biotinylated secondary antibodies and avidin HRP. And we were able in this way to analyze a large number of mice. We also had hoped that we could determine a time during the day when all the mice would have a peak or so, but that was illusionary. We just had to go at a large number and try to figure out. And here you see some of the profiles of individual animals that we got. You can see clear peaks. Sometimes this mouse there was either in a trough or was stressed. There was nothing. This mouse here was probably stressed. Other mice, we see clear peaks, but at the time of sacrifice, they are in a trough period. And then there were some that we get, unfortunately, as you can see, the secretion peaks are very short, that we got on that rising flank of the peak, which means when we then, about 10 minutes later, perfused the animal, it was on the peak. In fact, we got daily patterns in this way for 24 mice that we ended up analyzing. And of these 24, 4 were at the peak, which is a problem which also informs some of the future work that we're going to do, because it's a bit of a shotgun approach and you go through a lot of animals in order to get a few at the peak, but we need those for comparison with the trough ones. In any event, for this study, we had 4 at the peak. And then with that, we could move on to our structural work. And then now, before I get into the actual anatomical data, let me ask the question, sorry for that, why do we need super resolution microscopy for that at all? And I like showing this slide here when I try to answer that question, because imagine this be an image from a biological sample taken on a wide field microscope through a 100x objective using an EMCCD camera with small pixels, and you have the pixel size of about 160 nanometers. And the PSF here, of course, it's light microscopy. Let's assume it's about 250 nanometers in X and Y. So you see, at that level of resolution, you're looking at a single point source with a Gaussian intensity distribution around it in that field of view. Now, what if actually there's something smaller underneath? There's actually 2 points there, 2 point sources, but they're closer than 250 nanometers together, which would mean then they appear in a normal light microscope that's diffraction limited as 1 point source. That's why we need super resolution. And in particular, why do we use storming, or how does storm microscopy work, or which is one flavor of single molecule localization microscopy, and that term already gives it away. So what we do here in this technique is we label our structure, for instance, in that schematic here, these cytoskeletal fibers with an antibody, hopefully very dense. And then we label those with fluorophores, which have the property under certain chemical conditions, they are able to enter a dark state in their energy diagram, from which they come back, which is they go there, but they do not emit fluorescence when they do so. And then they come back from there in a stochastic manner, one by one. And over any field of view, you then take a large stack, a time stack of images. In each of these images, different of the fluorophores are on. For each of these fluorophores, which would look like something like that on the camera, you would determine the Gaussian distribution, the center of mass of the bright spot, and you use that coordinate for reconstruction of a super resolved image. That's the basic principle. I don't want to go into more detail here. Just let me show that little video from Ricardo Enriquez. It's already, I think, something like 20 years old. He published that on YouTube to illustrate the functioning of his software that he published there, QuickPalm, at the time when super or single molecule localization became a thing. And what he did there, he was a postdoc at Paris at the time. He went to the Eiffel Tower at night and took with his camera a time series of images of the blinking lights that are on the Eiffel Tower at night. He fed his algorithm with that, which would detect the bright spots and calculate the center of mass, and then reconstruct the shape of the Eiffel. That's basically exactly how any form of single molecule localization microscopy works. Okay, that's what we did in tissue. Specifically, we look at the region of the brain where the growth hormone-releasing hormone neurons are located, which is that tiny little nucleus here in the hypothalamus. This is in the forebrain. It's at the base of the hypothalamus here, the so-called arcuate nucleus, or ARC. And the data that I'll show you here are based on labeling for synaptic proteins of both inhibitory and excitatory synapses.